There you go. Okay, perfect. So if there's no other questions, I mean, you can always ask, like, if something comes up as we go through today's lecture material, we can always discuss it. So today we'll continue along similar lines to finalize the network X, and then you'll see some more applications and some more code. And then next time we'll continue with a different library, but along, because I want to have like a kind of a big section in the course on biological networks and network data structures in general, because that's very, as I mentioned before, a very general data structure, very useful data structure for bioinformatics data. But also next time, the one we're going to see another library, the spectral, we're going to start segueing into the machine learning aspect of the graphs. Excuse me. So graph theory and machine learning for bioinformatics continued, and I'm showing you here, in case you don't remember from last time, the slides, right? And basically, I posted both links on the Blackboard. That's the textbook version of the material that you can use to read. And today we're gonna also start working with a collab, which some of you might have worked, or you might have worked previously, or you might have played around after last week's lecture. And then, as I mentioned, that's going to be the main thing for our practice, coding practice, and the homeworks. And the homework is coming later today for being due next week. Okay, so we used and so a Python library called NetworkX, and I hope all of you had the chance. You'll definitely have the chance to solve the homework, to go read the documentation for that library. The link is here, and examined the last time different types of graphs, directed, undirected, weighted, and played around a little with the NetworkX, creating some of those graphs. And then you know, we just did some easy data structures, some sample data that we made up on the fly, just networks with a few nodes. But now we'll learn to pass real data sets. And actually, real data sets we'll see today, but I still keep it like in a few nodes network, so that's more easy to go through the lecture. But then for the homework, or you know, if you want to do further study on your own or research applications, you know, you'll be parsing networks with thousands of nodes, and that's where the power of this programming framework and any programming framework that you use for bioinformatics data shines through. Okay, and we'll do some more Python explanation of data structures and stuff like that, just to, for those of you who are already like very well practiced with Python, you know, it might be a little boring, but I just want to make sure that everyone who's, again, you need to know Python to go through this course, but you might be a little rusty. So, you know, we just explain some of the stuff, so you warm up with the language. Okay, so let's see. First, we talked about real data sets. So first, let's see some where we can get some data and some example data sources or databases, public databases where you can get real bioinformatics data sets. And that's what we're going to be using today, and also for the homework. One of those is the BioGrid, stands for Biological General Repository for Interaction Data Sets. And I'll go over each one of those in more detail. Another one is the INTACT. It stands for Interactions, from the European Bioinformatics Institute. And then another one is the Stanford Biomedical Network Dataset Collection, SNAP for short. So this, I picked three as example. If you go search out there, there's plenty those to be found. And then I would suggest to do that, just to get an overview of the field. We don't have time to cover all those today. But the reason I picked those three is one, that's here in the US, and it's very well known in the biological community. The other one is in Europe, obviously, and both those are backed by big institutions. And the third one I picked because it's more, first of all, it's not from a big institution. I mean, it's in Stanford, but it's made by an individual researcher in a lab like we are, for example. But it has very good collections on the data sets. And also, I would, and we'll go see whether the data sets are there. But I would also suggest go look at this research group's website, because they're computer scientists. That's why I also picked it up, and they do a lot of algorithm research for graphs. So you might find some interesting things there. So it goes beyond the just pure data repository. Okay, so first let's look at the BioGrid, and I link everything from the material. So this is the BioGrid database. So pretty dense there, and then I mean, you don't need to look at everything. You should have a look, some announcements they have, just to get an idea what the database is about. The things I want you to focus on is, and also for what we're learning in this course, is the types of data, right? So in the BioGrid, actually, it might have been in the US, but I think it might have been moved to Canada now. At least like the main PI moved in Canada and runs it from there. But I'm not 100% sure for that. Okay, so again, what I'm interested is a lot to talk about the data set formats. So how do we go find about that? Of course, there's the help for the website. And looking really quickly, I don't see anything here. Let me go to the downloads. Okay, so always the current release, and I see here a format called the My tab, and we'll talk quite a lot about this one. And there's another one called the Site tab. And when I say format, basically all those are zip files. But you see they have the extension My tab and Site tab and plain tab format. And that's like the whole database here. Big file. I mean, it seems not so big, like 140 megabytes, but actually those are highly compressed. So when you decompress, it might go even up to 1 gigabyte. And then they have also broken up the database in by organism, by project, and so on and so forth. Right. So this My tab and Site tab. Let's keep that in mind, because that's something that's going to come up at the center of what we're going to be talking today. And let's see if we can find the data extended database. Okay, so format details. All right, here we go. And I'm just doing that. I mean, you could do it yourselves, obviously, but I'm just trying to give you an overview of how you can approach those things if you want to get it, and you kind of have to do some investigative work to see what kind of formats, what they're available. Okay, so you see here this Sci My tab and BioGrid tab and all that stuff that we've been seeing. Let me go back to the downloads. Corresponding here, right? So XML, many of you might know, it stands for extensible markup format. It's kind of like one format that looks kind of the HTML. If you ever looked at the source of a website, it's all these tags and highlights. For us, it's easier to work with the tab-based formats. When I say tab, like a tab delimited kind of Excel like file, and I have some screenshots in subsequent slides. tab is all tab delimited. This is their own internal kind of tab delimited columns and rows, Excel type format. But the MyTab, the Sci MyTab, which is the one we'll be working with, is the one that's a standard in the community. Is an open standard format, and we'll talk more about it. We'll see examples with real files today as well. And this actually, I have the link in one of the next slides. We could also Google it now, just to go straight there since we're there. So it's an open source format, and then that's published on the GitHub. So GitHub, as you might know, it's an open source code repository, and you'll see this format has versions 2.5, 2.6, 2.7, 2.8, and I think it's I don't know why they don't have it here. Let me see if I have the right link in one of the next slides. It's actually up to 3.0 now, and each subsequent format has more information, right? And we can go really quickly over that. So again, it's like rows. Each row represents one interaction, and then each row has multiple columns, right? So if you go across one row, and again, we'll see examples in the next slides and the code. If you go across one row, you're going to find those columns, which the first one is identifier for interactor A, the first node, identifier for interactor B, the second node, which they are linked with an edge, right? So that's the bare minimum you have, and then you see as it's going further down, you have alternative identifiers. Those are basically protein identifiers. You'll see it's just an alphanumeric type of entry there, represent the protein ID from a public database, the NCBI, and I hope all of you know about what I'm talking about when I mention NCBI, right? The main bioinformatics database where all the data goes. It's the reference database maintained from the National Institutes of Health, right? Like where is the proteins here? We can leave it open actually to put an ID from our data just to demonstrate. Okay, so and that's what the official identifier from that NCBI database. And when we can also see UniProt, but UniProt is sort of the corresponding, because you see here NCBI protein. NCBI is National Institutes of Health here in Bethesda, Maryland. UniProt is from the European Bioinformatics Institute in the UK and in Switzerland. So there's and those databases mirror with each other. So those IDs are corresponding with each other, right? So you might see NCBI or you might see UniProt. Same thing. You can cross-reference those. And then alternative identifiers, aliases, and then I'm not going to go in much detail now, but you'll see more and more information. Some type of interaction types, which they take them from a control vocabulary. So let's see if we can find here interaction type, molecular association. So that's like a set of like fixed terms they use, right? Enzymatic reaction. So this is kind of a dictionary to describe the experiments they did when they figured out the protein interactions or the gene interactions or whatever, and they try to standardize the terms, right? So that's on that, and I'm just picking really quickly some fields, and that's where was that, the interaction types, right, from the dictionary. Source databases. This refers to, I guess, the database where they store the interactions, or the database where they store the proteins. The ones, remember when we were making the network the last time, we were putting attributes on the nodes, right? Whether it's a gene or a protein. That would be here, biological role of node interactor A, node A, and biological role of node B interactor B, which are connected with a line, and then some experimental roles, which we could attach as attributes in our network as well. Annotations, meaning like what type of protein they are, and you'll see in the files that I have. I'm not going to go into much detail during the lecture, but after the lecture and for the homework too, you can go there and look at those files to look at those data in more detail. And actually, it's interesting, like as I mentioned before, every subsequent version, an advanced version of the SciMyTab has more information. Like the 2.5, it doesn't have the biological roles, and you would need that, but the way that works is that you'd get the idea of the interactor here and go to the database where the NCBI or the UniProt, and then try to find the biological role there. And we can see an example of that today. But this is kind of inconvenient because if the biological role is in the file, then you can immediately parse it out with the Python as you go through the file and just attach it as an attribute to the node, right? And that's what we would prefer working with. SciMyTab, I believe, is over 2.6, which they start adding the biological roles. On 2.7, I'm not sure, but the latest you can get, the better. The interesting thing is that I suggest you do that as an experiment. If you go to the BioGrid, I mean, it says like MyTab here. Yeah, so you can actually see it on the screen. It's MyTab level 2.5. And I don't think the reason they do it is to keep their files small, because even if you went to 300 megabytes, I think it would be fine, given the data sizes. Today, 300 megabytes is nothing. But I think the reason they do it is like, if they were to upgrade in the newer format, they would have to pull from all those other public databases, the information for the biological roles and update all their files and stuff like that. And that's, of course, you can automate that with some coding and everything. But for them, it's more like the man hours they would have to spend, the employee hours, to make sure that everything is correct and there's no errors as they integrate the data. So I think that's why they didn't do it. But for us, it's not so convenient because, as I mentioned before, we prefer to have everything in one file and load it in our graph as we parse out the file instead of going to other databases. But don't worry, like the files I have is what we need. So that's one thing to remember for those databases. So everything is not perfect out there. Okay, so let's see really quickly the intact. So this is the European counterpart. And as you see, those guys, they use the MyXML, different website, a little different design. Somebody mentioned last time about the COVID interactions. You see here is kind of like front. And so you can go parse it out. This one actually is it's not of much interest to us as bioinformaticians, because this is basically showing what you would see if you parse out the file, right, line by line of the file. But for a biologist, which they don't do the coding and they want to go to the website and see the stuff, that's pretty useful. For us, what's interesting is where's the download to get the whole thing, right? To get the downloads. Where is the downloads? I guess FTP. All right, so let's see if we can open FTP in the browser here. Okay, so that browser doesn't like the FTP. Oh well. So we can. What was that one? Okay, that was a reference from before. Okay, so we can see it here actually. So you see they use the same MyTab, again, stuck at 2.5. And actually, they have the MyTab 2.7. And I remember now when I was preparing this lecture last week, that's where I got the data they're going to be using today. So that's if you download this one, it's a big file, and well, it has a lot of, if you unzip it, it has a lot of smaller files inside, and each one of those smaller files is going to have an ID, and that ID corresponds to the PMI ID, which stands for PubMed publication. PubMed is another database here from the NCBI, and basically it's the publication for this experiment where the network data was, where the wet lab experiment took place, whether it's a yeast-2 hybrid or a proteomics experiment, that they figure out the protein interactions, and then they put those data in a file and they publish the network after they make sure everything is verified. So you'll see here, let's say 300 files, and each file is going to have a name, and that name is the PubMed ID, which if you go here and search where's PubMed, if you search with that ID, you're going to get actually the publication if you want to read it, where the data was generated. But then, of course, what we're interested in is the file, where's the network data, and that network data is going to be in the MyTab 2.7 format, which, as we explained here, because we were looking at the 2.8, that's going to be this one. So when you'll be looking in that file, as we'll look later today, you're going to be seeing each row having all those, it's 42 columns. It doesn't look very pretty if you see it in Excel or just open the file. That's why we parse it with Python. But not all of those columns are going to be like full. Some columns might be empty. Okay, and then last, but do we have anything here? Let me see. No, I think that's the one that works best for us, and that's the whole database. I like this one because it breaks it by publication. And if you download and unzip that one, you'll see some files are big, some files are small. And again, we said one row of the file is one interaction. You see some files, they have like 3,000 rows. Some files have like 15 rows, and that means like in the experiment where the file was produced, and again, if you put the ID of the file on the PubMed, you can get the publication of that experiment. Some experiments, they were like high throughput proteomics experiments, and they were able to capture 3,000 or whatever that big number is, protein interactions. Some of them were simple, like yeast-2 hybrid experiments, which basically you do them in Petri dishes or whatever, and they have like 15 rows, right? So they were able to capture 15 interacting proteins. It's more like, not like they were able to capture only 15, it's more like that it's targeted to capture the interactions for one specific protein. And you'll also see that in one of the example networks, we'll see over in the Google Colab, where you see one, basically, you see the interaction is like one protein in the center and the other proteins like connected to it, right? And that doesn't have to do, that's another thing I want you to remember by informatics. It doesn't have to, let's say, correspond to the reality in a sense. It's more like how the experiment was generated with that capturing the interactions for that protein. That's why it looks like everything links to it, right? So there's a lot of what I'm trying to instill into you. It's like there's a lot of stuff that you need to subtle, things that you need to be aware out there, right? Because again, it's data sets, and they're generated from experiments, and experiments have specific designs which might lead in the data looking in a specific way, right? Which might be different from the biological reality, because the experiments is something that you design when it comes to the wet lab experiment and the protocol. And then we have the last but not least database, the Stanford one. And you can see here some really, really big data sets with 3 billion interactions. So this one definitely wouldn't work in the Google Colab. You need probably a computer with very big memory to work with this data set. But there are some smaller ones too. I mean, those ones are like, they have some ecological data sets, butterfly networks or whatever, but you can see here which ones are the ones who are interested, right? For genomics and molecular biology, the gene function, gene-gene interaction networks. These are huge data sets because the genes interact in so many ways in the cells, right? But those data sets, just to give you just also my experience from the bioinformatics, because they're computer scientists, it's the cleanest data sets you're going to get compared to the biological database ones, because computer scientists, they work a lot with algorithms and they want to have very deterministic stuff, but also the data sets which are the more abstracted from reality, right? So if you want to develop algorithms or have something clean to sort of like benchmark things, this is the place to go. If you want to do biological discoveries, you have to go to the biological databases, right? Like the Intact and the BioGrid, and there again, you have to be careful. That's why they give you the links for the papers, as we'll see. Read the papers, make sure you understand what the experiment is and what the data that you get represent, and then you can go mine the data for discoveries. And there's huge potential for discoveries because there's a lot of this data is generated automatically with high throughput experiments, and there's so much data out there that you can basically build a research program by just using what's in those databases, right? As long as you write the code and the algorithms and bring all those tools together that we're doing in this course, and mine those data sets for essentially for biological discoveries, right? And you'll see we'll be doing quite a bit of that stuff when we get to the machine learning aspect of the course. Okay, so this is what we have with the databases. I guess we also covered that slides, right, with the available formats and the official specification of the MyTab on the GitHub. We've seen it already. I guess here it links. It's again on the GitHub. It's a cleaner website. That's basically the GitHub pages, right? GitHub.io, which is also what I use to host the material for the class. And this thing we're looking before, that's the raw code repository, right? Which I also use, and it's available if you want to download the raw code for that website, right? Because it's an open source website. Okay, so that's the MyTab format. If you want to read it here again, I linked the cleaner version from the slides. I guess we don't need that anymore. Let's keep it clean. Okay, so we cover the downloads for BioGrid. We cover the MyTab. And as I said, most intuitive to use is the tab delimited formats like the MyTab, because we can think in rows. Rows meaning like iterating through, parsing line by line, as we'll see in the Python code, and then grab the columns and put each element from each column in one array or list in Python and parse it out very easily. That's why we like that, and not the XML. And as I said, we prefer at least 2.6 or greater version of the MyTab, because that has the roles biological roles of the interacting proteins, so that in addition to the structure of the network, we can put attributes like we did in the previous lecture for each node, what kind of function they have, right? And that's what brings the value when we build on those data structures, because you'll see again when we go to the data mining in subsequent lectures, our goal will be to I mean, we can get a network which has all nodes annotated and erase manually or artificially the annotations from some of the nodes, and then use the data mining to assign annotations there, right? So, but what you do in a real research setting is like you would get a network where you have some nodes with annotations, some nodes without annotations, and then use the data mining algorithms to assign annotations to the non-annotated nodes, right? And that's what the graph machine learning algorithms do. And then, yeah, because it's already building there, the annotations on the sci-MyTab greater than 2.6, we don't need to go to a different database and pull the data and integrate them and make sure that everything is integrated correctly, and so on and so forth. Okay, so yeah, before I show you that, is there, I guess I can put it up, is there any questions regarding what we discussed? And that basically is how you would see it if you open the file in the text editor or the terminal. Basically here, I grab the first, that's what this command is doing. I grab the first 2 lines of that file, and then the first line is the header. The tab delimited file, right? First row starts here, and I guess second row starts somewhere here, and each column has tabs in between, right? And these tabs correspond to the tab data down here as well, right? So if you just want to read it like that, you would see that this one, interactor A, that's actually where the second, so this is just a header, the first row, right? That's just the header, which describes what the columns are. Just visualize that. Here I have it in Excel too. So visualize that, and you can actually, if you download that file, which I have them all linked here, I have 3 files, and then you open it as a TSV in Excel, that's how you're going to see it, right? Interactor A, that's the first line, and Excel opens it up like the tab delimited stuff automatically. So that's a header line, first column, second column, and so on and so forth, right? And if you want to see it in the terminal, that's where it is, right? So that's the first row, and then first column, and then first row, second column, second row, second column, right? And so on and so forth. And then you see here what we mentioned, right? The official ID of the proteins from the database, from the UniProt, and then alternative identifiers from the intact database. And as I mentioned before, those databases, they all cross-reference with each other. That's why they have the alternative identifiers there. Now, this file, I got it from the one zip I showed you in the BioGrid, which has where was it? Was it, maybe it was the MyTab, which had, as I mentioned, all the files as correspond to publication, right? So this, and we'll try it now. I haven't actually tried it. This should be the publication ID. Again, if you download that zip file, it's going to have like hundreds of those in there, and each ID, each file is going to have like a network, which came from the experiment publication. So let's see, 146.05208. So let's put it in the PubMed. 146. My short memory is terrible. 0505208. 208. So as is, that would be the idea for publication. Let's see if that works. There you go. So what? So this one here is a Drosophila interaction map. So that's a network with the measured in the fruit fly, right? So you can go read the publication. So what does it say? 7,000 proteins, which have 7,048 nodes, which have 20,000 interactions, 20,000 links on them, and so on and so forth, right? So that would be all the information for that in here, right? And today we'll work with some smaller files. So I linked all the files here, and let's actually I'll go get them now because I need to upload them in the Colab. Download, as you see, I use like okay, save file. Let's save it locally. I'll get all of them very quickly. Okay, let's get that guy too. Save, and then I'll upload them into the Colab. Okay, good. I think we have it all. And then you will need to get those two or other ones I put in the same with the same method to work with the homework, right? All right, so let's go ahead and do that on the Google Colab notebook. So basically, I linked on the Google Colab notebook here. And if you want to do modifications, so that, let me clear all the outputs. Actually, I've done that before. I apologize. So it's more clean. If you want to use, I mean, you can view that code and run it, but if you want to modify it, you will need to make a copy under your account, which you'll be logged in. It's free if you don't have to create an account at Google, and then save a copy in Drive, right? So it will create a copy in your Drive. Now let's clear all the outputs here. Let's not, do I have more? Yeah, it's a couple more. Let me clear. Be patient there. Unfortunately, there's not a way to clear all those outputs. Actually, so the way I do this, I used to work. I mean, I work obviously now on the Google Colab. I used to work like exclusively. Now it's more like for showing for the class. I use command line and Vim and the interactive Python. You know, interactive Python, IPy. That's how it's called. And I do all those things in the terminal, right? With the terminal text editor. So it's faster to work with that. Okay, so that's the Google Colab, which you make a copy. And then, as I mentioned the last time, if like I've been working at it, I mean, you can work across computers or the same computer, but it doesn't really matter if you close the window. I mean, if you close the window, open up again quickly, this doesn't happen, but if you close it and come up after 5 minutes, the virtual machine, because those are virtual computers, which you get at Google, it shuts down, so you lose the file, so you have to re-upload it, right? And the way you upload the files, you go here, and I think I put a link, file save copy, right? So I have a screenshot here, but I'll show you live, but you can refer to the slides if you need. Okay, so let's go to our downloads and upload our files, right? And it reminds you what I'm saying, like files will get deleted and runtime is recycled, means like if you step away from it, they recycle it. So you see that one was a big one, so it takes a little bit of time. Was that the one I was using here? Well, that was a different big one, but anyway, I picked up a big one, so it should be fine. Okay, so they're still uploading, but anyway, for those ones that are here, basically the way you do it, right? Copy path, and then then the path you can paste it in here, right? Or have me demonstrate copy path, and then you know, you could put it here, right? Doesn't matter. Okay, so remember those are the code cells, right? You can hit code or text cell if you want to write some text as a note because it's interactive notebook, and then adds a cell here, and you run it with the control enter or the play button, right? And you can delete the cell here, and then always remember to import your libraries. And again, when you walk away from that thing and they recycle the instance of that virtual machine here, you have to rerun everything. So if you have imported here or created a variable with some data, it's not going to work if you go run a cell further down, because the code is there, but once this virtual machine has shut down, everything is lost from the memory. So whatever variables or imports or whatever you initiated here, if something further down depends on the stuff up here, it's not going to work, right? So you have to go again and run it from the beginning. Okay, so was not authored by Google. Yeah, so that was authored from my other account, and I trust my other account because it's me. And then we can go ahead and run it. So I imported this one, no errors. Right, it shows green. If it was an error, it was have told us. And yeah, so I'm gonna do a few cells here, and then we'll take a break, and then we can go in the rest of the code for the second part of the class. So any questions regarding to what I mentioned here? The uploading or those files, or how you get the links or anything like that. Maybe some of you already are familiar with the Google Colab. So if you have any questions with the Colab, I mean, we didn't have homework last week, but you know, I pointed to the Colab, and I hope like most of you went and played a little around with it if you're not familiar. But any questions on the Colab?
Sorry, yeah. What are those commands? The cut command and the WC command?
We'll go into that.
Oh, okay.
Yeah. So, but in general, if you have any questions like how you access the Colab or anything you're confused, because that's going to be like, I mean, again, you're welcome, as I mentioned in the last class, to use your own IDE. Even though with the big data sets on your own computer, you're on your own on that. And also like to install dependencies and everything. But if you don't do that, then our main platform is going to be the Colab. That's why I want to make sure everybody for our lectures, for our homeworks and everything, make sure everybody understands it. Okay, so if there's no questions, let me show you one the question you just asked. One trick with Colab, because again it's virtual machines and it's Linux virtual machines, you can actually run Linux commands. You can get a full terminal, but here you have to sign up with the monthly plan, which starts at $5 or something, which we're not going to need, so you don't need to do that. Despite not getting the full terminal, you can actually run some Linux, Unix commands by writing the Linux Unix command and preparing it with an exclamation mark. And that obviously is not Python. And then from your CS 122 or 132, where you did the Unix stuff, hope you remember those. If not, you can just put like, and if you don't have a Unix system, you can say man cut. Well, this one might bring like a barber shop or something in New York City, but man page, I think man page cut. So those are the manual pages for Linux. So you can read if you want the parameters or they do, or you can just Google and say Unix cat command how to use or whatever. There's plenty of stuff. If you worked in a terminal, you can say man cat, and then this is the official manual page that would come up. I'll explain to you here. So basically, what this is doing is when you have tab delimited files or comma separated files, you can parse out sections of those files. So essentially, what I'm saying for this file is -f is which fields, meaning which columns. So get column 1 to 2 and column 17 to 18 and column 21 to 22 from this file here, which is the file I uploaded, and copied the path, right? And then I paste it in here, and then Unix pipe, and then to the head command. Again, you can look at the manual pages. For Linux, head command is print the first lines. There's a command called tail, print the last lines. By default, if you just use head, it just prints or tail the first 10 lines for head, the last 10 lines for tail, but you can also customize it and say print the first 4 lines, right? So let's run this and see what's happening. Okay, so here it is. So I grabbed, and I'm not going to go see the whole file, right? Because the whole file is going to look like this, right? So actually, let me leave my slides here, and then if I want, so that's from the last time, if I want to refer, I can use this one, yeah. So that's going to be my whole file, but I'm just getting the first, the second column, and then 17th column, 18th column, and then 21st column and 22nd column, which, as I mentioned before, is the ones which are most interesting for us, right? Basically, the first 2 columns is the IDs, which we use to build our network, and then it says which databases those IDs come from. Unfortunately, here, and I didn't run out of time when I was looking for files. You see here, you get the other columns, 17 and 18, where we're interested in biological roles, which is the ones which would attach to the nodes of the graph. So we have some known nodes, some unknown nodes, and we can do the predictions with the machine learning. But unfortunately, this file, if you see all the roles here, is using the standard MyTab dictionary that we've seen before with the biological roles, but they all use the ID from the standard dictionary 0499, which is unspecified role. So, and they basically measured some interacting proteins, and they know there are these, but they don't have the annotations to them. This might be an artifact because, again, they didn't integrate that file with the annotation databases. So everything's going to become as unknown here. So the network going to be created is not going to have so much rich information. I mean, we have on the interacting types all protein, because it's a protein-protein interaction network. So we could attach that to each node. Say it's a protein, like remember we did last time. Some nodes were genes, some nodes were proteins, right? Here we could attach like to every node being a protein, because that's across all the lines of the file. But it's fine. I think we're just more interested to see how to build a big network. Now, if I wanted to say, let's say I'm curious to see like how big is that file, right? So so there's the WC, which stands for word count, another Unix command. Hope you remember those from your classes. So let's see how big is that file. And if you do man page WC word count, you'll see it has an option. Let's do it. Man page WC, right? There you go, Linux man page. So L counts how many lines, right? So let's count how many lines in that file. So it's 16. So it's actually going to be 16 rows in that file, a small network, and it's going to have 15 rows, which are actually proteins, right? Because the first row is just the header, right? How about, let's see. So this one is actually too big. It didn't upload. Oh, well, maybe I should find another file to put there. Okay. So this one, say, has 105, so 1 for interacting proteins, so a little better. But I think we're going to stick to the small one because it's better when we do the visualizations, right? I mean, we can try the big one too if we have time at the end of the class. Okay, so I don't need this cell here, so I'm going to stick with that one. Okay, and let me show you the last Unix commands really quickly before we go to the Python, and then we take a break. All right, so what I want to do here is basically just work, let's say, with the first and second, first and second column. So I can actually, since we have on the 17th and 18th that's here, we have unspecified rows. I don't want them, so I'm just going to keep the first and second column from that file, and then again my cut for the 2 columns, and then I use Unix pipe. I'm going fast over the Unix commands because supposedly you know or remember those from your 132 and 232, or you can practice those as needed, as not our focus here. We're just using those as tools. Okay, so get first and second column pipe, which means the data that come from the cut. And if you wanted to see it, I can demonstrate it for you here. I mean, it doesn't hurt, right? So those are the 16 columns, right? You see this one actually it's missing something from the file. You'll see how that's going to come up in the network, right? So this part of the command, I'm going to pipe it to the set, which is serial editor, right? And I'm using the regular expression here, which you also hope remember from those classes, which means serial editor dash E expression. So what's the expression? Substitute that much uniprot KB. That's my regular expression, right? You have to put backslash because in the regular expressions the double colon has a meaning. So match that and then substitute it with nothing, right? And do that globally. So basically the cut prints out line by line, and then it pipes it to the serial editor, the set, and then the serial editor executes a regular expression on each line. And that regular expression, what it is like whenever you match this pattern, substitute it with nothing, right? So if we are to execute it now, which means like this first part, which I printed here, it's going to get piped into the set and processed with that. So that's what you're going to see. So you see, it cleaned it out. Of course, you could leave the uniprot KB there, and as you load the file with Python, clean it out with Python regular expressions or Python string slicing and things like that. But I prefer to clean with the Unix tools and then you know, get the IDs ready and cleaning the file to load them, right? So that's what we have after this command, and this one is just redirection to write to a file which is called content/network TSV, which is the file we'll be using to load with Python. So let me actually run that too. Right, this one doesn't have an output because the content is getting redirected to that file, and this one I can delete it. We don't need it. And then you'll see now, yeah, this file is written here. And then let's examine it. All right, I'm just doing the first 10 lines, which you see here. the output, right? So when we saw it before too, when I explained the command to you. And then here we do the counts, which is like 16, right? So count the line 16. So 15 interactions, so 15 links, 15 edges in our network to construct after the break, and 15 because we have one the header as well. Okay, so our file looks good. We have it, and then we're ready to parse it. Okay, so that's with the Unix commands. We have the files. We're ready to go in and load in Network X. So let's please let me know if there's any questions on those. We can answer before the break, which will be for 10 minutes until 11:25. And then we could come back to continue the remaining of the code. So is there any questions? Go ahead.
Yeah. Are you going to be sharing this specific file through Collab with us, or do you want us to download it from the website and do it ourselves?
So if so here's the link, and if you click on that link, basically, I mean, when I clicked it, I was logged in, right? So I had immediate access. You're going to see something which will say this is from a third-party person, and then you can only run it, but not edit it. So what you would do if you are logged in your Google account in your browser, you're going to say set a copy in Drive. So you're going to get your own copy under your own account in the Google Colab, and then you can edit and do whatever you need with it.
Oh, okay.
Yeah. And then if you use like locally or in another cloud service, you know, like the binder, you know, because if you are like really into big to open source and like Google, anti-Google, why is it not loading? This is an open source kind of Google collaboratory, right? And you might have your own instance of Jupyter, which gets those IPy and B files locally. So you can also download it and run it locally, right? But of course, remember, if you run it locally, you will have to install all the dependencies, right? NetworkX, NumPy, and all that stuff. Where that's the advantage of Google. It's built in. Actually, what I'm trying to do in that Jupyter book framework that I'm using, I have activated an option. It's just when I compile the code in the book, it doesn't appear here. There's an option to, when you have like code blocks, and I see the copy here, to just get a link directly to open it in the binder or the Google Collaboratory. So essentially my goal would be like, just go in here and just click, and it just opens on the Colab, but I haven't figured out how it works, but I'm just linking the whole Colab, and then you can make a copy and run cell by cell as we do here. Okay, so I hope, did this answer your question?
Yes, thank you.
Okay, perfect. And then any other questions? I guess we can leave the outputs.
Can you briefly explain again how, like with the regular expression, it's able to take just the numbers and not the like before the colon?
Yeah, no problem. So the regular expression, let me zoom in so we can see it. The regular expressions is unique. It's a kind of a language on its own, right? There's so essentially, if you learn regular expressions, you can use it to any language, right? Every language, Python, Java, whatever, it's going to have you know, a function where you say put the regular expression in, and based on the regular expression, it's going to modify the data accordingly, right? But this regular expression, and I'll explain to answer your question, like how it's structured. It would work in every language. I mean, when you run the Unix command, the Unix, the terminal is the bash, right? The interactive shell, which is a programming language on its own, right? But you could get that and use it within a Python construct. So the regular expression, the universal language of regular expressions, for this specific example here, so you have the first part between the first two slashes, which means the pattern that you match. And then the second one between the second set of slashes, which is the pattern which you substitute with. And then I'll explain the other stuff too. So pattern that you match is UniProt KB that we see here. And then it has a colon as well, but you couldn't put it like that because in the universal language of regular expressions, this has a special meaning, which I don't recall. What is it now? I mean, I know that single character would be like matching any character. So if you want to specifically match the dot, if say, for example, this one, instead of colon, it just had a dot, right? And you would say specifically match the dot, you need to backslash it or to escape it, meaning that because the dot has a special meaning in the universal regular expression languages, meaning dot match any character, but you want specifically to say I want to match a dot, you want to escape that special meaning of the dot matching any character and say I just want to match specifically the dot, right? And the same with the semicolon. I think the semicolon means like a range of characters or something. So, but we want to match specifically the semicolon, ASCII character. So that's why we escape it here. So basically what that means is match exactly the UniProt KB with a semicolon afterwards. And now when you match this, then just substitute it with nothing. I mean, we could say substitute it with hello, right? That wouldn't help us because we don't want to insert a hello in there in our IDs, right? But we say substitute it with nothing. That's why when we match whatever is between the first set of slashes, which is the one we want, then we substitute with nothing on the second one, right? So that's how the regular expression works. So basically, always the regular expression is like first two slashes and then last two slashes, right? And this one is it means substitute, and this is called modifiers, and this one means globally. So globally means like you working on a single line, right? Because the cat, it prints out lines and it just passes them out through the pipe, which is that one here. It just passes them out to the set and it works line by line. But globally means across the line, and means that if there's more than one occurrences of that pattern across the whole line, and we see here there's two of them, just do the exact substitution, match and substitution to both of them, right? So there's two occurrences, two matches of that UniProt KB colon, and then both of them, because of the global operator, and even if there were like 3 of them and 100 of them, it would be the same thing. We would just replace them all with nothing, which is the second part of the regular expression. So similarly, think about if you wanted to substitute that with also with nothing, because say you want to clean it out, you would just put that thing here, and remember to escape your colon, which is a special character, right? You can say, if you're interested, you can say like learn, regular expressions in 20 minutes or whatever. So you can say like bunch of stuff here. I guess this guy is using Python. And then I can also show you escape, character, regex, regular expression for sure. So there's like tons of stuff out there, right? So you can go Google around and see what's that stuff, right? And the colon should be somewhere in here, right? So you see the plus is a special character, and so on and so forth. right? So some characters are special characters, right? Okay, so I hope this answer your question. Please let me know if there's
Sorry, one more question.
yeah, go ahead, go ahead.
So I'm trying to do the copy path in order to get the data, but I can't seem to get it. It keeps saying no such file or directory.
So you see, let's try to debug it like this, and everybody else, like you can go ahead to the break. I guess let's take the break to the until 11:30 because I will need a little break too. So everybody else, feel free to do your break. We'll come back 11:30. So you see the file here?
I don't actually. I don't see the same thing. I see some other stuff.
Have you uploaded the file? So the file that when you open the Colab, the files are not going to be there. Even when I close mine and open it again, the files are not going to be there because it just cleans it out basically.
Right.
The code stays there, and you might see the outputs too. But as we said, you're going to have to rerun the code from top to bottom. But what you need to do is to where did I have the files linked? So you need to download those files by clicking on those links locally on your computer, on your download folder, and then just do file, upload. You see I have downloaded those files here. So maybe you didn't see the window that popped up. So did you download those files on your computer?
I'm gonna download it right now.
Just
That would explain the problem I'm having. Yes.
Yeah.
Yep. Give me 1 second.
And then file upload. Yeah, the one is big actually. So I realized now it has a problem uploading. But try the two smaller ones. And then do file upload, and then it should appear here. And then you can do the copy path. So let's try that. And if you want, I can give you screen permissions if that helps. So you can show me what you're doing. So you just have screen permissions now. Let me pause the recording. Okay. Is there any other questions you might have thought during the quick break? Or should we go ahead and do the Python and there's no questions? Okay, so if there's no questions, then we can go ahead. All right, so now what we want to do is parse out those files. This file, at least with one we work, and remember, we're just working with a sort of very lean version of the original file, which are parsed out with those Unix commands and some regular expressions that we've seen. And again, those regular expressions, you could have used them in Python and did the whole thing, like pick out the columns and clean out the columns in Python. But I'm kind of like, I guess, old school too, and I know the Unix stuff very well. So I cleaned up the file and made it these nice two clean columns with ADs, because as we explained, there's not really much else in that file. I mean, there's the biological roles, which in that one it's all unspecified. I will try later today to find, because in that huge zip file, there's like hundreds of them, and I'll just picked up really quickly 2 or 3, and it just happened those ones were unspecified at all. But I'll try to find one which has biological roles that are like entered there with some real biological role, which you could use in the homework to make a network with annotations on the nodes, right? But for now, what we have in that clean file, which we're going to use with Python, is the IDs, right? And each row, as I mentioned before, is one interaction between interactor A and interactor B. So that would be the one node, and that would be the other node, and that means there's a link between them, an edge between them, right? And then that same node, for example, here has a link with another node here, and this node 0.14.9.20 has links with other two. And of course, when we parse those out with the network, with Python and network X, and then we visualize it, you'll see how nice it will show. And of course, this one is a small network, so you could do it by hand basically, but the whole purpose is being able to do that with files which have like 16,000 nodes, right? Which I'm not going to do here because the networks become very dense. So I'm interested to show something clean and small, right? Okay, so our Python code, oops, sorry about that, for parsing out the file, enlarge a little bit. So it's just open a file handle, can read up on the Python stuff if you you don't remember those concepts. So open, but it's pretty straightforward to see here from the code too. I open this, the full path of the file as a read file, and then S means as a file handle F, right? You could, this is basically a variable, like every other variable in Python. You could name that whatever you wanted, right? You could name it like file hand. Of course, you have to use it like, and you see here, when I enter the variable, it's saved into the kind of memory of the instance of the Google Colab where we have. So automatically it pops up, so you know we can select it, right? Just one convenience it has. So open the file handle, and then I name that file handle F, or name it file handle, or whatever. So for each line in this file handle, so it's going to iterate, meaning going one by line. Then that's another Python here. So for each line, so it's going to go on each line. So it's going to grab first, very quickly. So first it's going to grab, which we don't want, and I'll show you, because we don't want the header. So I'll show you how we can exclude that in the next code cell. But first it's going to get the first line, then it's going to get the second line. So for each line, so grabbing line by line, stored in each variable, this variable here. So when it gets, for example, to the second line, this whole thing is going to get stored in that variable here. And then that variable line that you see here, it's right here. So what do we do with that? We do an R strip, which means I remove the new line. So this is Python methods, right? You just chain them together, one method after the other. So remove the new line. Here doesn't matter so much, but if we were to like print out where we parse, then you know we wouldn't want those extra new lines there. But it's always good to do R strip, remove the new lines, and then do a split, meaning split the line. So that's, say we're on line 2, R strip, remove the new line from the end, and then split it based on a delimiter, which so split is a method, right? We chain together, and a delimiter, which we give as parameter to that method, because the methods always get their parameters in parentheses. Here, this R strip, it doesn't get any parameter, but the split, it gets a parameter, which is the delimiter, and we specify the delimiter being the tab, right? Because it's a tab delimited file. And I think it might have worked even if we didn't specify it, because I think the tab is a default delimiter the split works with, but I'm not sure, right? So you're not sure, you just put it just to make sure that your code doesn't error out. Okay, so let's say we're line number 2, we're stripping the new line, then we're splitting with the limiter being the tab, and then also what we do, I think in that file actually it doesn't make a difference, but that was from the previous one, which I haven't removed. So for example, like if it was the other fields, 17, 18 that I had before, right? So, you know, if we had those 2 additional fields, so this is what I'm doing here, say, strip the line, split it in tabs, so you would get like 4 fields, and then get up to the second field, not inclusive, the second field. So it will be the first field being the 0, the second field being the 1, because remember we measure 0, 1, and up to, so that's the 0, that's the 1, that's the 2, and that's the 3. 0, 1, 2, 3, the fields, but get the 0 field, the 1 field, and up to 2. Without the 2 is not inclusive. So basically, if we had all the additional columns in the line like I had it before, just to get the first 2 columns, right? That's what this thing is doing here. So basically, it cleans the line, it splits up in chunks, its column, and then it's going to put that into a new array with its element of the array. I call it array because I come from the Perl world. The right way to call it is list in Python. And each element of the list is going to be its split that we did, right? And the reason to tell it to put in a list is because you put in the brackets here, which is a list annotation, and then that whole list is going to be stored in the pair set. I could have named it something else, right? So let's go ahead and run the code, and you'll see it here. This is the pair set list, and you see, of course, it grabbed the first, which was the header as well. So this is basically a list of lists, right? So the first element of the list is a list, the second element of the list is a list, and so on and so forth. And the elements of the first element, which is a list, is, you know, the first 2 fields, right? Which is, let's see it here, like this one, and then also this one, right? The second element of the list, which is also a list, has 2 elements. The first element is this one, you see it here. Second one is here, and so on and so forth, right? I could print the whole thing if I wanted, right? So that's my whole file, basically a list of lists. And if I haven't done that thing here, right, just as an example, well, now it's a little confusing, but you would see, like, again, list of lists, and each one has like the first one, the second one, the third, and the fourth, right? Let's see it here. Just to understand. So you see, let's go for row 2, second list in the list of lists. This one, this one, this one, this one, and then this one here, down here, and then this one here, down here, which I don't want. That's why I say, I put that one and say get up to the second element, right? And there you go. So I cleans it up, and then I see a question: why does one not have an identifier? Could you maybe, because I don't understand.
There's one of these things that starts with Q. It doesn't, or oh, it doesn't have a Q, or no, it just has a dash.
You mean why this one has a dash? Because it's an error in the data file, I think. They didn't put an ID for that one. So it's just on this file. you're referring to that one, right? I have highlighted. Yeah. Yeah, so this is just an error that just didn't put the product and ID. I would actually be interested. I mean, this is a small network to do data mining on. But that's basically what we're going to be doing when we get networks where they have some roles here. We're going to be going and deleting some, not their IDs, but their roles, and then using the data mining to assign roles to the to those algorithms there. Yeah, and then this one I'll print I can print, like, say the second to the fifth of the file, right, without the ID. But again, you'll see that we parsed out the header too. So when I go, and I'll get back to that for questions, but let me do the next cell as well. When I go do my network X, right, the typical thing we saw before, initiate a graph, and then add edges. Remember, let me go to the slides here. I guess it's the same thing that I explained. We can leave it here, that's fine. We'll use it as a recapping. So remember when we were doing the graph edges, remember when we were building networks the last time, we were first adding the nodes and then adding edges between the nodes, but we said, like, you can essentially skip adding the nodes part and just put an array which has the node identifiers and just use the add edges method, and automatically it's gonna build the graph both in the nodes and the edges. So that's what we're doing here, right? We're just throwing in this array, the pair set, and then basically has the node IDs, which is however many unique node IDs the network X finds. It will create a graph with those nodes, and then each pair of nodes, that's going to link them with an edge, with a line there, right? Now, if we see on this pair set, we parsed out the header too, and we don't want that. That's why we tell it, get the pair set to build our network, but starting from 1 and to the end of the pair set, right? That's what I have down here, because we just want to exclude that sort of junk in there, which is the header IDs. I mean, it's not junk, it tells us like what the data is, but we don't want it when we build a network because that's not obviously a nodes, right? So we say start from 1 onwards, right? And then we said about this one, what's happening here. And then the last line, which we've seen before, draw our graphs with the labels, right? So let's go ahead and do that. And that's our graph, as you've seen before, with the node IDs as labels, right? And everything came from that one. And then you can verify it, that this is correct. And of course, remember, each time we redraw it, because it's using a random layout, it's the same graph exactly. It's just like flipped around, right? You can do that as many times as you want until you you verify that this is the case. Okay, so we parse the file and the graph. So let me stop here and ask if there's any questions. I mean, this would be pretty straightforward, right? Because we did the last time. The only thing we introduced, we also seen it the last time, we said, like, as I mentioned now, just put in the pairs of nodes and automatically it's going to create the graph and the links between them. So you don't need to add first the nodes and then the edges. The only trick here is that we got this array from up here, which we parsed out from the file, and we just made sure that we exclude the first line, right? So we passed in the array from line 1 to whatever the end is, right? This had like 15 rows here, the array 15 entries. You might have had 1,000, 10,000. I mean, 10,000, you wouldn't see a clear graph here. It would take some time to draw it, but there's no limit here. But anyway, if you have any questions for that, let me know. But is there any questions still for that one here?
I have a question about the graph.
Mhm. Scroll down.
So where is the rest of the graph? Is there a cutoff point somewhere?
So are we missing nodes?
Yeah, are we missing nodes?
Which one?
I only see what is this? This is 6 nodes? Isn't it
Wait.
the graph is drawing between every node, right? Every pair is a node.
So this is the nodes, and this is the edges between them. Are you confused because there's more rows here than actually shows here?
Yeah.
No, but the thing is that you see this one here, for example. it
Oh,
has
I see.
linked-
They're repeats. Okay, yeah. The pairs are branching off. Okay, I see it.
Yeah.
Sorry.
So yeah, so this guy here has like 3 links, right? This one has 2, and this one has 4 links.
Yeah. I see it now. All right.
Yeah.
Thank you.
So and you always expect to get like smaller than the number of rows, right? Okay, and
The nodes
go
have a
ahead. Go ahead.
what does the dot mean with one of the nodes? Is that just the starting point, like in the graph?
This one.
Yeah.
So this one, it comes actually from that one here,
Oh, I see. Okay.
which is an error in the data file, which is an empty dash. So it should have been an ID. So that's just an artifact. Okay. Any other questions? Let me go back to my slides too. And the slides, basically what I said. I'm just like it's notes for what I said. I mean, you can watch the recording. That's how I would study, and then have the slides next to it, and just go point by point as a note, as notes, and looking and understanding the code, right? But I think we don't need the slides now, as I explained in the code. Import network X, and remember that we cannot directly. This is, I got it from last lecture, right? That we're just throwing in there the list with a pair of nodes, which is that's what that is here, right? We're just throwing in the list with a pair of nodes. Okay, I think this one, ignore it. This is an error. I'll fix the slides. This came from the next slides, actually. Okay, good. So let's go make some more networks. This is actually the network. Let me rerun it now really quickly, since we have it here, but then I have to change it. Let's do like the 1500 very quickly. So I want to show you the other one so you can see it alive. All right, so the head we don't need. I'm just basically recreating the array, and let's redraw the network with that. So you see how this network looks? That's what I have in that slide. It's what I mentioned before. That's what is in that second file, which is an artifact, right? Because basically, I mean, it's a valid experiment, but the way the experiment is one of those like fish experiments, I think that they call them, where you put a protein and then see what binds into it, right? So it's true. It's those interactions are true interactions, experimentally verified. Again, you could put that 1500 4283 to the PubMed and get the paper and read. We don't have time to do now, but my guess is that it's an experiment where they use like a protein as a bait to see which other proteins attached to it. That's why these data are like that, right? So again, that goes back to the bioinformatics data out there. It's from experiments and the way the experiments are done. You might have things like that. That's why you need to refer to literature. Don't think like you discover like a new pattern in the cell or something, right? This is like how the data was generated by the experiment. Okay, so let me redo it with the other one. It's just going to take 1 second. Because the other one is more reasonable network. And then you play around in the homework and on your own as well with other bigger networks and more variable networks. Okay, I'm just rerunning the same thing over. Okay, so here's our other one. Okay, more reasonable. Okay, so some more. So we explain about those ones, and let's talk about connectivity of the graph nodes, connectivity of the graph, and this is all like graph theory. Basically, that plays in when you have like really big networks, right? I mean, now 16 nodes here is like nothing. We have thousands and thousands of them. You need to use algorithms which produce metrics to summarize like how well is connected your graph, the density of the graph, and also things like query. How can I go from one side to from one node to the graph to the other? Meaning that are those nodes connected through a path, right? Starting from one node and then following a set of edges and going through some intermediate nodes to get to the other one, right? Which I guess I don't need to say, but in a biological network, that's like it has many implications, right? One might be depending what kind of network it is and what kind of data you representing. It might be signal transduction, right? How you know the protein that's outside of the cell receives a signal and gets a modification. Then this modification cascades through additional interactions with other proteins, you know, to a protein in the nucleus and starts transcription of a gene, right? And other changes in the cell. Or it might mean that you have like gene interactions, right? 3 or 4 genes, one regulates the other, and so on and so forth, or metabolic interactions if the network represents a metabolic network. Which this path through the graph, it would show like how you go from the initial metabolite, complex carbohydrate, for example, to follow some, because you can represent networks of chemical reactions with graphs and follow some steps, some chemical reactions, and convert that metabolite, that complex carbohydrate, to a simple sugar or ATP or whatever, right? So this stuff you can do with those algorithms, and there's algorithms which summarize the connectivity, there's algorithms which query those paths in the graph, and all those things are in Network X. I've put also the links on subsequent slides. And also these algorithms can be used for research questions, right? For example, if you have one mutation, right, meaning like one gene becomes dysfunctional, which meaning like you would remove that node from the graph because it's dysfunctional and doesn't contribute to the signal transduction, or it doesn't contribute to the to performing the metabolic step it's supposed to be performing. And then you want to see if the graph breaks down, right? If, for example, when you remove that node, the path, the initial path from node A to node B that passed through that node that you removed, if it's still feasible to go through other nodes, right? Because, like for example, just making it up now, you could see if you remove that node, you can still get from that one to that one here through this one, right? But if you removed that one, for example, then the graph gets disconnected. There's no way you could get to that one or that one with a weird dash ID here, right? So this one is kind of like a mini hub of the graph, right? If you knock this one out with a mutation or whatever in the cell, then those ones would be disconnected, and there would not be a way to get from this node to those two, right? And again, whatever this graph represents, right? It might represent a metabolic. I mean, this one is obviously a protein-protein interaction network, but if it was representing a metabolic network, that would be like this enzyme here was mutated So it doesn't work anymore. So there's no way to convert that complex carbohydrate to those simple sugars, right? And then, you get some disease or something, right? Like, dementia or whatever, accumulation of complex amyloid carbohydrates in the cells or whatever. And if it was a signal transduction pathway in the cell, it would mean like the signal couldn't get from interacting proteins to the final destination, right? So the graph breaks. So these are all those things that you can do with those graph algorithms, and you see basically the role of the bioinformatics is to just get the data, model them in that data structure, and then model them correctly, whether it's a metabolic network or whatever else, and then using those algorithms when you have the data structure properly set up, answer those biological questions, right? What happens if I remove this node? And we're not going to be doing like a lot of this stuff in this course, like we do a little for the homework. My focus for introducing the graphs is, as I mentioned before, moving a lot into the machine learning. So the goal is to answer questions of like mining the data and assigning function to the unknown data, right? It's not going to be questions like I mentioned. What if this node gets mutated? Do we still can get to those other proteins, right? To those other metabolites. But this is two things you can do: the data mining or searching the graph out of the many things you can do. Okay, so this one, number of edges per node. Okay, so let's see the connectivity of the nodes, some simple graph metrics. Before we go there, a couple of functions that I'm showing you here. So basically, you can get a list, an array of all the nodes in the graph and store it in that one called nodes, and then get the first 10 elements, right? That's another function we have. But just get into those graph metrics. So the first one we're going to do is just a general information on the graph and the degree histogram of the graph, meaning that degree is, when we say degree of a node, means how many links a node has, right? So this one here, it would have 1, 2, 3, 4 links. This one here, it would have 2 links. This one would have 3, and this one would have only 1, right? And a degree histogram is like a histogram, where you count how many you have for each one, right? So basically, this one would return, and we can run it, and I can explain the code. It would return like how many links you have for each node, all right? And before I get there, I use this function, which is built into the NetworkX, which is called info, and the variable that stores the graph, which is G for us. And basically, it just prints out that line. It says it's a graph with 6 nodes and 6 edges, right? So I would say always do that. If you parse a big file, you could probably tell by looking at the file how many edges it's going to have, but you load in NetworkX, and then that's much cleaner to do. So use the info graph, and it shows 6 nodes and 6 edges. Now, the other function that starts, we'll see in the next cells, a little more interesting functions that starts get like even more interesting. Is like, let's get a degree histogram. And you see here how I do it. I do from NetworkX, import the 2. So those are under the function, so the NetworkX, and I import the 2, the info and the degree histogram. I mean, you could say like from NetworkX classes functions, import everything, right? But I like to keep it like that to know exactly what I'm importing for the specific operation that I'm going to do. It also saves memory, even though for this type of operations here, we don't really make a difference. But when you have like big programming libraries and big data sets, and you do import star, that can have some impact on the performance. But I like to do it here for code readability. So degree histogram, the built-in function, applied to a graph, and that's basically what it returns is an array which has the number of links, the number of edges for each node. But what I like to do is also, and let's see if you go over it with Google. Let me delete that because that's annoying. If you go over it, it just tells you like what after we run it, it just generated a list with five items, right? So you can see it like really quickly. And I like to sort it, so I apply to that the sort function with reverse order. That's a built-in function for lists in Python, and then I just print it, and then that's what I get here. And just a little tip to see. So that's as we said, like that's a nice thing with the Google Colab is you just that variable is initialized, so it comes up. So you can when it comes up, like see first I do DG, and then it comes up, and then I don't have to type the whole thing. I can go down, select it, and then press enter, and it autofills it for me. And then another trick, if I hit dot, because it knows, as I hovered with the mouse before, and it showed me it's a list, right? So it knows that internally. So basically there, if I hit dot, that list comes up with the little cubes, which means methods. So it shows for the list, which it knows, again, it's a list, it shows which methods are available, right? And you can go down and select the sort, right? So you can either, if you know, which you probably do, this is simple stuff, Python lists, you can apply the sort method. But if it was like some other object which you don't know it very well, you can use that functionality of the collaboratory to really quickly, instead of having to go to another website and look at the Python documentation, to really quickly see what the functionality is. And also, once you put in there, and again you hover over with a mouse, it can show you really quickly what the parameters that it takes is, right? And you see that key none is if you use like dictionaries and stuff, which you don't use here because the sort applies to dictionaries, I guess. But you can use the reverse option, which by default is false. That's what equals false means. But we're gonna do true, because we're gonna reverse it from the node with the largest number of links to the node with the smallest number of links. And let me see, I'll be able to do it now, but there's a way when you hover the mouse, it gives the documentation full on the right side. Let me see. Well, I can't do it now, but that's fine. Okay, so yeah, so print the information for our graph. We call the method degree histogram. I'm sorry, the function from network X degree histogram on a graph. Got a list with the number of links, which the official term for the number of links per node is degree of the node. That's why it's called degree. And histogram because it gets for all of them. And then that list we sorted in reversed order, and we printed it, and that's where it is. You don't see the node IDs here, but you see that one has 3, which is this guy here. Another one has 3 too, so it's a histogram, right? So in the histogram, you just group together by category. So this one is grouped with that one under 3, and then there's the guys with 1 and 0. And that's like very simple statistics. to see, how is your graph structured, right? We're going to go now into some more interesting ones, and that starts getting to the more advanced graph theory concepts, which again, we're still scratching the surface here. One being the centrality, which is a fancy name, but again, it's kind of the same thing with the degree histogram you've seen before. It's just like normalized for the size of the graph, and that takes into account that some of the nodes, if it's a very big graph, they might be connected to thousands of other nodes. That's the hub nodes that we also discussed before, but it's a much bigger scale if we have bigger graphs. But essentially, this degree centrality is normalized for the scale of the graph, if you will. And let me run the code here. So the reason I'm not very interested in that metric, but the reason I put it here is more for the Python coding practice and understand the data structures for it, because what it does here, well, there's two things. One is that it also gives me the node IDs. So before you saw, it was just a histogram, right? It was grouping together, saying like we have a bunch of nodes with 3-degree connection, and then nodes with 1-degree connection, and so on and so forth. Here, whatever that normalized metric is, and when I say whatever normalized is, however it calculates it. Actually, if you go to the documentation of network X, it gives you like the exact mathematical formula that it calculates that. But for us, we're more interested in the coding here. Whatever that metric is, this number, it gives you specifically for each node, right? And again, that metric, what it is like what's the number of connections the node has in normalized to the size of the graph, right? That's what this metric is. Now, because we're going to have the ID of the node 2, what returns is the dictionary. Here's the brackets, and dictionaries in Python and every other language is key value pairs, right? So key is the node ID, and value is the metric, which is the centrality, right? So node ID and the value. So what I wanted to do, again, I wanted to sort it from the one that's most well connected to the one that's least connected, right? So here's some interesting Python coding for you. So this is my dictionary, right? So basically what I do is I do for key value pair, that's what this is in the dictionary, and then I basically reverse it, and then I do the sorting based on the value, right? So we don't need to go into too much detail for that, but basically what I'm saying is get my dictionary and just look at the values, and whichever one sorted based on the numeric values, and sorted in reverse order, right? And that sorting operation, so this is the opening, you see it highlights the brackets, opening and closing bracket for the sorting operation, and inside the sorting operation we get the key values of the dictionary, and reverse it by assigning a value key pairs to be in tuples in an elements of an array, because you see now here we have an array, and then we sort that in reverse order, right? So just to make a long story, and then we print it, to make long story short, is that we get an array of tuples, and then you see this node here, which has 0.8 connectivity, followed by this node here, which has 0.6 connectivity, followed by this node, which has 0.4. And then you can, it's a small graph, you can verify that. Here's looking at the 014920, right? That's the most well-connected node, and his normalized centrality connectivity is 0.8, right? Biggest than everyone else. And then he should be followed by those two guys, as you see here, right? That's their IDs, right? The 9836 and the GK3, which is right here, and so on and so forth, right? And the reason that being here, and if you had like a graph with thousands of nodes, you wouldn't be able to, I mean, you would be able to draw it, but you wouldn't be able to visually see what's going on. And that's, you would just be thinking on code and algorithms. That's where you would use that stuff and, get out like the most connected proteins you have, right? You want to see like in that network, I have in the cell, which is the protein, which is like really a hub, right? The most connected. If it gets mutated, the whole network will break down. And again, we're just scratching the surface with those algorithms. But from what you see here, even in a research setting, if you applied that, you wanted to find the most connected protein, that I think that would give you, enough to go by. Of course, if you really wanted to answer more specific scientific questions, you would have to read like the tens of algorithms for graphs and the ones that Network X implements, and go apply those there. And I guess this is the same thing. I have it twice. I don't know why. So we can skip that cell. Okay. So is there any questions for this one? Again, don't get hung up too much on the code here. Just take it as it is. More like interested for you to understand the concept. Of course, you're welcome to study this Python idiom in more detail if you want, because this stuff allows you to write quick code. So let me know if there's any questions in those concepts here.
Especially on the coding, just in, yes. I understand the concept, but just in general. So if you go back to the code, why do you have to set the key to none? Like if you set it reverse equals true, wouldn't it automatically do it? Do you have to set it to none?
So the key to cell is tested. You mentioned to just leave it empty, or?
Yeah, like why do you need to set it to none?
Let's see, that's a good question. I don't remember when I did. Let's see, key. Well, we removed the reverse. Let's see. I guess it works the same way. I might have actually been doing, if we read the method for sorted, let's see if we can get it quickly here. I might just have been putting the default actually. So the key equals, yeah, you see here, key equals none is the default basically. So I just type in the default, so it doesn't really make a difference, right? Mm-hmm. But essentially also to answer your question is like, if you wanted to sort based on key, meaning that you would specify a specific key. So let's see if we put like this one. So either, in my case, either put in none, which is a default, or put in nothing, it's the same. But what if I put a specific key? Let's see what happens. String number is not common. Okay.
I think it wants an anonymous function there, like a lambda. So like if you wanted to sort, like say like on the, or maybe not anonymous function, but like length. Like I think if you put in len, I think if you put key equals len, that would work, except they're all the same length. So then that'll just,
let's see.
Yeah.
Like that. Yeah, there you go. So essentially in that case, key's length means that it's short based on the length of the string. That's the key, something like that, or how would that work?
Yeah, like len, it'll just be when it gave you the not callable area, it's because it wants the function there. So it'll, whatever you put there, it'll call that function on, I mean, given that you have like value and key in a tuple, I actually don't know exactly how that's working because there's two things. Like, I don't know what, like, I don't know exactly how length is working, but you could put in any function there. And then it'll call that function on every item that's being sorted.
Okay, so essentially that allows you to customize how your sorting based on key happens by even writing your own function or using the built-in functions. Okay, that's very interesting. All right, so that's another good thing to know, which it might come handy because sometimes the bioinformatics data is quite messy, so the default sorting might not work, and it's good to have that power of controlling what happens in that sorting or whatever other operation you do. Okay, so what's the next one? Connectivity. Okay, so essentially this one returns so this connectivity measures how well connected is each node with all the other nodes, right? And you can think here, because we're going to report for every node how well connected is with every other node. This is going to be, especially for larger graphs, it's going to be a bigger data structure, because essentially we get each and check for all others, then the next one and check for all others. So it's going to take, not here because it's a small graph, but in a bigger graph, it's going to take time to compute, and then the data structure is going to be big. And what it returns is the first node as a key and dictionaries as value. So we'll see it when we run the code. And then its value in the dictionary is another dictionary which contains key values, which is the, if say, let's see in our graph, we pick up this node here and run this connectivity metric, which we will do, then it will return a key being this node, and then a value being a dictionary, which will have key value pairs. So the value is going to be a dictionary, which is also going to have key value pairs. And in that dictionary, you would see this being a key, this being a key, and this being a key, and this being a key. And then the connectivity metrics being, I think it's, I don't remember the definition now. I'm tired also after lecture for 2 hours, being the distance. So let's print it. Yeah, so that should be. All right, so let's examine it specifically instead of just me talking out of the air. Let's find the 0, 1, 4, 9, 20. Okay, so let's see where it is. So let's find that key. So you see, okay, here it is. All right, so then, so that's my key, and that's my value, which is another dictionary, as you see from the brackets. So within that dictionary, that's the value. We have more key value pairs. So there's the Q99836. So let's go find in the graph. Q99836. And then it shows a 2 for that one. And this one, Q9UGK3, it shows a 2. And then the rest ones, it shows 1, right? So for the 1511 and P259 and the dash, it shows 1. Okay, so for those ones, it shows 1. For this one, shows 1. And for those shows 1. Okay, so without reading the definition, my guess, and then if you read the definition now, please let us know what you see there. My guess is that the closer it is, the higher ranks it gets, because you see those ones, which are immediate nodes of the one we're examining here. So that's our key, that's our value, and then it just basically the dictionary, which is the value, has all the other nodes in the graph as key value pairs, and the closest they are, the highest the value they get. That's what I see from here. And let's examine another one. Let's examine the 0, 1, 5, 1, 1. Okay, so where is the key for that? And let's see the key for that, 0. Okay, let's go here. So you can see, right, this is the keys in the first level of the dictionary. This is why, okay, here I see it. Okay, so that's our 0, 5, 1, 1. So basically, it says Q99 861. Okay, so this one has all one. All right, so I understand now something different, that actually these numbers here, it means the number of different ways you can get to the other node. So our Q, let me actually redraw the graph down here, so we don't go up and down. Sorry about that. We'll get better at that as time goes. So let me, I'm showing you the trick now how you could do it yourselves too. So let me redraw it here so I have it here. All right, there you go. Perfect. All right, so this one is here, right? So that's our key. And then our value is the connectivity, is a dictionary which shows connectivity with all the remaining nodes, right? So then for that one here, which is our key, it shows that it has connectivity 2 with that guy here, right? And connectivity 2 means that there's 2 ways you can go to it, right? And then with that one here, that's our key. With that one here, it shows 1, so there's 1 way you can go to it. Then with that one here, so from that one to that one, right, it shows again 2 because there's 2 ways to go to it. And then for that one here, it shows 1 because I guess there's 1 way to go to it without like overlapping again. And the same for that one here. So that's what the connectivity measures. And let's not get too much hung up onto it. I mean, you can always read the definition because we're not doing like graph theory here in the sense, but it's more like for the applications to bioinformatics. But what I want you to focus also a lot is and ask questions if you have how this data structure is for those of you who are still warming up into the Python, right? So again, we have keys being the node and value being a dictionary, which also has key value pairs, which shows the connectivity of this node with every other node in the graph, a metric for the connectivity. Okay, so is there any question in this concept? Again, don't get hung up a lot in what that connectivity means or how it's defined or whatever. It's good that we went over it just to discuss it. Okay, so if there's no questions, let's do the remaining ones. Okay, so there's 2 more algorithms I import from the network X. Let me go to the slide, and then here again I would suggest follow the links and then go to the definitions. You see I have links for both the algorithms and the functions, which in the homework I will ask you to run some algorithms and some functions, so you're going to have to read the the definition and know how to run it. But besides just finding the specific ones that's requested to the homework, I would say like just have a quick, or if not so quick, like a good look to all the algorithms and functions that are available there to show you what you can do, what operations and statistics you can get from the graph. And then I see something on the chat which says show how mRNA, virus, different end results, antibody production, different vaccines. I'm not sure what this, Henry, what this refers to, like the, does this refer to this metric here? Would you like to tell us a little bit more?
Yeah, I think it can be used for all the matrices that you mentioned, either give degrees of freedom as well as the weighting. This will lead to maybe a more direct path as to what antibodies you want to produce in using particular vaccines. Okay.
Okay, that's great. That's great comment. I mean, there's a lot of biology in there too, I guess, how the vaccines and the antibodies work. But you know, if I was to summarize it really quickly, I guess it's how you're going to cascade, how fast you can cascade the signal, right? What's the
to produce the correct antibodies for the different Omicron variants and Delta variants.
Yeah.
Because you want to be specific. Yeah, using this path, you can find the quickest possible path to get to the right antibodies, and how you can combine different vaccines to get the right end, the right end product. Okay,
good, very good. Yeah, and that's another way, another domain you can model the data as a graph. And as we said before, right, you can model metabolic networks, you can model even like ecological networks, right, interactions within species. But when you put everything, of course, you have to model it correctly by knowing the knowledge domain, which here the one you present is the antibody design. And then when you model the knowledge domain correctly into the data structures, then all the algorithms is exactly the same, right? Of course, then you still have to think like whether the algorithm that you apply makes sense for your data domain. But again, the big gain here is like when you have it in the data structure, you have you know, at your fingertips algorithms to do operations on very easily and on large data sets, right? That's a power we gain from the programming and those libraries. Okay, so let's go see a couple more. Again, I refer you to the links, you know, to go read more details what they do. But essentially, these ones, simple paths. Basically, it just lists ways to get from one node to the other node. So you specify the node, you specify the graph, of course, and then you specify the beginning node, the ending node, and then it's going to return, well, what it returns is an iterable object, meaning a collection, a set, if you will. But what I do, so when I call the all simple paths method, that's the method, as you see here, that pops up. Another nice thing with the Google Collaboratory, pops up the documentation of the method, which says, you know, specify the graph, specify the source node, specify the target node, and it's going to return a single node, I guess, if there's no path, or an iterable object of nodes, meaning a set of all possible paths, right? And in my case, I put in another parenthesis, casting it. And that's one term you could use as a list, so I can print it, right, in a clean way. And essentially, let's see, here it is, the 936, the K3. So it just gives me like the two paths that I have, right? The one being straight from the 99836 to the 9UKG-K3, right? And then the second path is doing that cycle is from the 99836 to 014920, and then to the 09UKG3, right? So those two paths. Now, Yeah, so there's another algorithm which says cycle basis. So essentially, this one finds cycle paths in the graph, essentially like closed paths like that, cycles. And then you just give it as parameter the graph G, right? You can see, let's see if the documentation pops up. Oh, there you go. A list of cycles which form a basis for cycles in G, blah, blah, blah. A minimal collection of cycles, essentially to find all the closed cycles in the graph. I cannot think of a biological sort of analogy now. Maybe I'll think and post it on the material. But essentially here you see there's only one closed cycle in the graph from those three nodes, which I printed, and then here it is, right? You can see their IDs. And again, this method returns an iterable object, which you could cast it as even as a tuple, or maybe you could put in a dictionary too. Again, this is Pythonic idiom stuff. So if you're really into Python, it's good to discuss beyond the point and the time allowed it here. So we just cast it as a list and then we print it. So we get the cycles and one cycle because it's a small graph, right? But if it's a bigger graph that we're going to be seeing with bigger files in the homework, then you're going to be getting more of those, right? Okay, so now a little more algorithms, and let me go back to my slides. So we covered that. Yeah, the casting returns a generator or iterable, either way you want to call it, and that's a way, as I understand it, in Python to save memory and instantiate a list or tuple, whatever you want to cast it, and populate it with data when needed. Now, Hamiltonian and Eulerian, this is famous mathematicians which came, that's where the names come from, which came up with the algorithms for those graphs. But essentially, Eulerian means that you have a graph where you can visit one edge, every edge exactly once. And Hamiltonian means that you can, I'll show you here as examples in the picture, means that you can visit every node exactly once without stepping on the same node twice, right? So for example, this graph here, I have like a link to the YouTube video where I got this image from, if you want to watch the whole thing for more details. So this graph here is both Eulerian and Hamiltonian because you can go start from here, cover that edge, cover that edge, cover that edge, and you don't go to every edge. You go to every edge only once, right? And it's a Hamiltonian too because you can hop on that node, hop on that node, hop on that, hop on that, and then back to where you started without double hop on any node. And now this one is not Eulerian because if you are to cover every edge, you start from this one, cover it once, cover it once, cover it once, cover it once, and then, okay, so you cover the outer ones, then you cover that one here, but then if you are to cover that one, you're down here, so you have to go for that one, which you stepped that, we're talking edges now, you stepped on it already, so it doesn't work. You would step on that edge twice, and you can try like the whole day, find different ways and see it's not possible, right? So there's no way you can cover all the edges, we're just talking edges here, without stepping on at least an edge twice, right? So that's not Eulerian, but on the other hand, it's Hamiltonian, right? Because Hamiltonian, you just check the nodes, so you can go from this one to this one to this one to this one, cover all nodes without stepping on a node twice, right? Now that one is, and let me see if I can figure out the right path. Yeah, it's Eulerian because you can do that edge, and then that edge, that edge, that edge, that edge. So you cover all of them without stepping in a single edge twice. But it's not Hamiltonian because then you'd start from that node, go to that node, go to that node, go to that node, go to that node. But then in order to go back to where you started, you have to step on the second, on the middle one twice. And in the Hamiltonian case, not allowed to step on the node twice. So Hamiltonian again looks at nodes and Eulerian looks at edges. So this is not Hamiltonian. And this one, and let's keep it here, is not Eulerian, not Hamiltonian, right? So again, for Eulerian, you just look at the edges, the blue stuff, stepping on each edge once. And for Hamiltonian, you look at just at the nodes, the red ones, stepping on each one once, right? And yeah, that's the definitions here. So these algorithms is built in under, I guess, Hamiltonian tournaments and Euler algorithms is built in on the network X, right? And then if you see definitions of the methods, it says returns true. So basically this graph that we have is not Eulerian because, you know, it's pretty simple to see it actually. If you started from here, you travel that edge, that edge, that edge. And then if you are to cover this edge, because remember, Eulerian, you have to cover all edges, but stepping at each one only once. So you cover that edge, that edge, that edge, and then this one, but then you have to step again or the other way. You know, you basically stuck. You have to stop to step, you know, at least more than once to some edge. And then the Hamiltonian path, it just gives you what is the path on the graph to get to all the nodes, touch upon every node without stepping on a node twice, right? So what does it give us? Here's an array. It just gives it as an array of elements. So it starts by the, where is it? The Q. No, this doesn't seem, yeah, this doesn't seem right to me unless it starts from the 0, 4. It doesn't seem right because if I was to get the 09836, let me see here, and then cannot connect to the 01511 because there's no edge to that, right? So I'm not sure how this would work into a Hamiltonian path, but even if it reads it the other way, no, I won't read it. G to direct it. So I think this algorithm is not correct. But if you got the, I mean, the output is not correct. The algorithm is correct, I hope, as they have it implemented in network X. Yeah, if you understood the Hamiltonian and Eulerian definition, you would see that this one is not right, first of all, because if we were to read it this way, there's not a link from the 99836 to the 151. So there's no way you could hop from one node to the other. So we'll leave it at that for now. But the key point here is, again, from the biology perspective, however you want to model it, right? Like I would think like the Eulerian makes more sense if you were to say signal cascading, right? How can you go and see that information flows across all nodes of your network, meaning that, you know, you step on every edge of the network, meaning that you're transmitting signal from one point to the other by connecting every edge, stepping on each edge without doing it twice, right? That would be the ideal structure of the graph. And from biological perspective, that's like optimization through evolution, right? You have a graph which is signal transduction graph, and it's Eulerian, meaning that it enables you to go from cover all nodes of the graph without stepping on an edge twice, right? Optimized structure for the graph. Sometimes it's good from another biological perspective to have the replication, because that means redundancy if you have mutations and things like that. So either way you want to see it. Okay, so no problem. Yes, we're almost done in 2 minutes, but whoever, I took attendance already, so whoever wants to go to your next class, we only have one quote cell to do. Feel free to do so. And then last cell, we have, remember the adjacency matrix that I mentioned in the previous lecture, right? How you represent rows and columns, right? So you put the nodes in the columns and the nodes in the rows, and wherever you have a link, you put 1 and everything else is 0. And you'll see here that this is a symmetric matrix because it's an undirected graph, right? And actually, we'll talk about if you don't put the 2 array. Let me actually, can I print that? Let me print real quickly. That's the, that's a sparse notation. It's something that saves a lot of space because it's a lot of zeros in there, as we mentioned. And we'll explain that. It's pretty intuitive to understand this data format. We'll explain that when we get to the spectral library in the next lecture. But you know, I just wanted to do it to an array so you can see it as we had in the previous lecture, which is more intuitive. And yeah, so don't worry. I see you in the chat. Don't worry about the attendance. Yeah, I screenshot it before, so everybody was there. No problem. It's fine. We'll do the turn on the camera next time. Okay, so that's what I had for some of the algorithms. Again, that's really very scratching the surface. And you know, we're not doing graph theory here. You know, but of course you have to follow the links, and for your benefit and also for doing the homework, read the documentation for the functions and algorithms for network X. Go beyond the what's requested in the homework if you want. And then my goal is to let you understand how we can parse those files, load them into a network, and then once we have them there, we can think of what kind of biological questions we need to answer and what algorithms we can run in the graph, or statistics, metrics, centrality, whatever, to answer those questions. And as a last sort of bonus, I want to show you also that the network X has this, but it's good that we did it with the columns and parsing the files and everything. If you have just the 2 columns of the nodes in a file, you can just read it directly, not make any arrays or anything like we were doing before, and just make a network, right? So you'll see here, I'm just basically cutting the first 2 columns of the of my file, and then I just use this method which I import from network X, which is called ADJ list, and then this one just basically reads the data straight from the file and just creates the graph with just 1 line. There's a small problem here. If you remember, we haven't cleaned the file from the first you know, row, which had the headers, and you'll see that's going to appear in our graph. So that's kind of a problem, but that's a good way to read the stuff faster. But again, what we showed, I left it on the end because what I wanted to show in the beginning is the whole thing with the arrays and the regular expressions and all those things. So which comes very handy, you know, if you want to clean up the files, which definitely is going to come up in almost all the instances you'll be working with is data from these databases, right? So the easy way is there if you can do it, but it's better to know the full functionality and the full story behind it, so you can work with any types of files you will encounter. And I think that's everything that I had for today. I know we ran a little later because it was quite a lot of material, and I tried to explain in detail, and I had questions. But I have another meeting. I need to go in 7 minutes. But if anybody has questions for the next 5 minutes, I'm happy to explain, no problem. So please let me know.
Did you say we have a homework due next week?
Yes, I will post it by the end of the day or first thing tomorrow morning, and then it's going to be due in a week. And basically, we will examine some of the the concepts we discussed through a quiz. That's going to be the first part of the homework. The second part is going to be some coding, which is not much different from what you see here, but I'm just going to give you another file, which is going to be a little bigger, but not big enough that it cannot be parsed, not too big that it cannot be parsed by using the collaboratory. So if you understand this code and these functions, you should be able to do the code and the homework as well.
How do you want us to turn it in? Is it everything's through Collab?
So in the syllabus, it was specifying that, and we discussed that in the first lecture as well, that in the Google, some people they put links in the Google Colab, like when you create a copy and put your code in, or create a new Colab and put your code in, you can put the link. But my experience is that sometimes you don't give the right permissions and everything. And when I have 50 homeworks to correct and 10 the links don't work, and I have to contact the people, it's very difficult for me. So better put a screenshot of your complete Colab. I mean, we can do more than one page's screenshot, right, if it's long, but send a clean screenshot, right, not with your errors and everything. I mean, as you develop the code, you will get errors. So when you have the final thing without the errors, what I want to see, like here, right, you have the cells with the code and the correct output with the code working correctly, and that's the screenshot you sent as an answer. And the quiz, I'm actually going to use another online quiz which is offered from one of the databases we visited. So this one, you can send a screenshot showing that the quiz was completed, and then when we have written questions in the homework, just write it in a Word or whatever, and just attach that along with a screenshot with the code. So
that's all due together, right?
Yeah, so the homework is going to be due in 1 week, and then you say I'll put it on the Blackboard, and then you can attach the files there and submit it there as well.
Okay. Is it due next Wednesday midnight or Tuesday midnight?
Yeah, well, no, usually Wednesday. I usually put by the time of the lecture, so by 10 a.m. Wednesday, because the reason for that is it makes the students try to do it before then and ask questions. Because if I put it midnight Wednesday, for example, like people do it after the class and then they have questions, and then we did the class already, so that's the thing.
Gotcha.
And we have the discussion forum, for any questions. So I'll be checking every day. Just don't ask questions like 12 hours before the homework is due, because probably there's not going to be an answer.
Gotcha.
Yeah.
Thank you.
No problem. Any other questions?
I had a quick question. So for the adjacency matrix, is the idea that if it's like a 0, then it's not connected, and if it's like